since my Panopto recorder on my computer isn't working, and I haven't heard back yet, although I just put in an email request to see if I can get it fixed. I'm going to do a trial, short little Panopto with my phone, see how this goes, because my intention was to do this like my other Panoptos for lecture, but we'll have to be doing these for lab, and so we'll probably have some videos we'll make to show you guys how to do some things. Although, for this lab period 8 coming up, when you get back, well, not when you get back, when we resume things on March 30th, week of March 30th, so this will be our first lab back. There will not be a quiz, because there's no new material to quiz you over. You just took a test way back before all this stuff happened. So these are the, so you need to stay up with this. I think, like usual, your PowerPoints are going to be online, all that stuff's on Blackboard, all that. If you have a lab manual, here's the thing, if you bought a lab manual, you're probably in better shape than if you hadn't had one, because you're going to have to do a lot of stuff on your own, and having some kind of resource is going to be really helpful. Some of you have the electronic copy. There may be, I know a lot of these publishers are sending out emails to instructors about, making things available for free, probably sending them out to students as well. If they're not sending those to you, let me know, or I can look into seeing what kind of emails they've sent me to make things available for free. I know at first they were kind of like trying to get us to use their platforms that would have, I thought, ended up costing you guys money. We don't want to do that, but if they're offering stuff for free, we might as well use it. Anyway, so here's what you're going to need to be reading up on. Again, it's going to make, going to be really important to be reading those PowerPoints, reading the relevant sections from the lab manual. Period 8 covers primary isolation media. And there are five different types here. I've talked about, I believe, all of these in lecture. I know at least 4 of these, probably all 5, I'm pretty sure. So you should know quite a bit about them already. Well, you may not know that much about them, but you've heard about them. You're going to need to study these things. And there's going to be a lot of this information throughout the semester, the rest of the semester, on your quizzes and your next lab practical. Lab exam 2. So the types of media that you would have been inoculating, and I think Nyan and Fawn and myself are going to do, I hope, if we can still go to campus tomorrow, make a video showing you, what kinds of, this is simple, you're going to be doing, you would have been just doing spot inoculation, but we want to go over the results as well, what you should be looking for if you were to really get to do this. The types of media are listed here: PEA agar, which is phenylethyl alcohol agar; mannitol salt agar, also known as MSA. You've got McConkey, MAC agar, eosin methylene blue, which is EMB, and then blood agar. And all of the relevant pages are listed there. You would have been working individually on this. As I mentioned to my lab section before all this started, one of our last labs, I said, second half of the semester, there's more material to cover, but less actual work involved, I guess, in terms of physically doing as much stuff, because a lot of it's going to be, or would have been, gosh, gotta get used to saying that, inoculating and then incubating and reading results and recording results, and then eventually applying that to carrying out an unknown project, which we still have to work out the logistics of that. I've covered this in really, really good detail in lecture, so make sure you know all the things on this slide. I'm not going to walk you through and read every little detail to you, especially when it comes to things like knowing before and after, or not before, for and against, what things are selective for and against, what the different types of media are. PEA medium is selective for gram-positive microorganisms. It's the phenylethanol that does the selection. It's going to kill off, or either kill or inhibit, I don't remember which, but it prevents gram-negatives from growing but allows gram-positives. Primarily used to isolate staph and strep bacteria from samples with mixed microbiological flora, particularly bacteria. You'll divide your plates in half, or would be, this is the standard way of doing this when we're doing these tests. You're not streaking for isolation, although in reality, if you were doing this in an actual mixed sample that would be given to you, you would do this, you would do a streak plate and look for isolated colonies and pick out the ones that are representative of what would have grown and possibly be your actual causative agent from that particular sample. So you would divide the plate in half, and then for this one you'd be using 2 different microbes: Serratia marcescens, which is gram-negative, shouldn't grow on PEA; Staph epidermidis, Again, you would just be doing a spot inoculation on each side of the medium or each half of the medium. You incubate, come back and you look and see what grows. On this plate, on the picture of this plate, the result that you would expect to see would be this right here. On the right would have been the staph epidermidis, because that's a gram-positive. It's the one that should grow. Serratia marcescens, being gram-negative, would not be expected to grow. Mannitol salt agar. All right, MSA, selective for Now this one's a little bit more specific than some of the others. It's selective for Micrococcus and Staphylococcus. It utilizes 7.5% salt, NaCl, to select these salt-tolerant types of microbes. That's a pretty high concentration of salt. Most things kind of prefer, like normal saline, which is, I think something like 0.9%. So this is a pretty hefty amount of salt and is not very hospitable for most bacteria. It's differential for mannitol fermentation. That's the phenol red indicator is going to turn yellow if mannitol is fermented to acid and producing acidic, low pH. Now on this one, you would have been dividing your plates into thirds and spot inoculating with 1 gram negative and 2 gram positives. And the 1 gram negative, which would have been down here at the bottom, that would have been where E. coli was spot inoculated. And then you have your 2 gram positives. Both are staph bacteria. One Staph aureus, that's on the right. That's the one right here that is that is yellow looking, because that's mannitol positive. Staph aureus is a mannitol fermenter. Staph epidermidis is not, so it's going to grow, and it's probably going to look kind of red just because it's taking on the color of the medium. And so those are the expected results. You're going to get gram-positive, not gram-positive, you're going to get the Staph aureus and the Staph epidermidis, both staph types of bacteria, or if you'd have used Micrococcus, it would grow on here as well. So those are going to grow, and then the differential property is whether or not it ferments mannitol. MacConkey, I've talked about this before, so I don't want anything I've covered in lecture, make sure you go back and review that stuff. This one, again, you would have divided into thirds. Now, on the right, notice how you get this particular very, very kind of dark pink, reddish color for mannitol. This one's lactose fermentation. So the differential property is looking for lactose fermentation. Commonly, this medium used for isolating members of the bacterial family Enterobacteriaceae. Selective for gram-negatives, the crystal violet and bile, which is present in this medium, will allow for growth of gram-negatives, but not allow for growth of gram-positive bacteria. Lactose fermentation, the neutral red, is the pH indicator. So now on the left, when you look at that one on the left, so you've got, this would have been inoculated with 2 gram negatives and 1 gram positive. Staph epi, gram positive, isn't going to grow down here where it was spot inoculated. The other two will. E. coli is a strong lactose fermenter, as you should remember from lecture, and so it's going to have that really bright pinkish red color there on the right. The Serratia marcescens is gram-negative, but it is not a lactose fermenter, so it's not going to change color. Now, this one kind of looks like, unfortunately, I can't think of a better description, it chewed bubble gum that's kind of worn out its flavor and color, so it's kind of pale color just because of the color of the medium. It even looks a little bit yellowish, but that's not really significant of its fermentation of anything in particular. In fact, this is not fermenting lactose, so it's a lactose negative. EMB, same particular use. It's mostly used to isolate fecal coliforms like E. coli. Same selective and differential characteristics. They're just going to look different. This one has E. coli, Serratia marcescens, and Staph epi. Now, it looks like there's a little bit of growth, and maybe that's just where it was spot inoculated. There shouldn't be any growth of Staph epi since it's gram-positive and shouldn't grow. On the right is E. coli. And you can kind of see on the outer edge of the bacterial growth from the spot inoculation kind of a greenish sheen appearance. That's typical of a strong lactose fermenter like E. coli. Serratia marcescens will not turn color. Sorry, it's going to look just like it did on the previous one. Here it looks a little bit more red or pink, but it's just because of the pigment that the bacteria produces. So it's lactose negative. E. coli is a strong lactose positive fermenter. Blood agar, oh my gosh, we've talked about so many things about blood agar. You should be pretty darn familiar with blood agar. Enriched with 5% sheep red blood cells that are intact. It's differential for the types of hemolyses, which you should know already, and you will be tested and quizzed over those types of hemolyses. This one was inoculated, spot inoculated with Staph aureus, which is a beta hemolytic one, Strep viridans, which is alpha hemolytic. It's going to have kind of a greenish brown appearance, which that's the one on the bottom. And it's hard to tell on this one, but if you hold these up to the light, you'd be able to see right through the Staph aureus growth, or at least around the growth, because of the hemolysis of red blood cells. Staph epi on the left, just that white growth on the left of the plate, that has no hemolysis whatsoever, so that's gamma hemolytic. So I'm about to wrap this one up. I don't want these to last too long, because then they become difficult and cumbersome to upload and too lengthy for you to really watch in a reasonable amount of time. So I'm going to kind of keep these short. The next lab will involve, now again, for your so after your first week of online instruction, the following week will be a lab quiz over primary isolation media. Then the following week's lab will be where we get into biochemical testing, and then you would have been doing lots of these. So we're going to have a lot of results to talk about in the next video. Try to figure out maybe how to break that up maybe into 2 lectures, or 2 videos, because that may be a lengthy one in itself. I don't know. Haven't done this before in this manner, so I don't know how long that would take to cover all of that. There would have been an assessment about how to determine what your contamination is, if you do have such, which, again, you're not getting to do this, but you need to be able to we'll have to probably give you a scenario where we give you a plate and say, well, okay, you inoculated with E. coli and staph and maybe even strep, and it was a PEA plate, and only the gram-positive should have grown, but you got something growing on there that shouldn't be. Your E. coli spot shouldn't have grown, right? But something's growing there, and then because, there shouldn't be something growing there, well, it's contaminated. It's likely contaminated with either the staph or the strep bacteria. Now, again, based on some properties of staph and strep, you should be able to figure out which one it is. We'll have to address that and how we're going to assess that. But be aware that's one of the additional things we're going to have to deal with is these activity assessments that you are routinely doing in lab, and how to compensate for that, how to get your participation points. One thing that we're going to do in A&P is give you some little questions about things in the videos. We're not going to make these videos just because we like making videos. I kind of like it. It's kind of fun. It's different. But we expect you to watch them. And so one thing we're toying with is putting some little tidbits of information in our videos. And for participation, we'll ask you these questions that say, maybe it's something we can add to the quiz, or some way put this out there that if you did in fact watch the video, you'll know the answer to some of these things that we covered in the video that maybe you can't necessarily just read online, something that we specifically talked about. I have to give that some thought how we'll implement that, but something to kind of motivate you to make sure and watch the videos. Just like in my lab, as those of you that have me for lab know that I expect to have you show or demonstrate things to me for participation points, because I know people will tend to skip out on those things, and I know that's going to be the case with these videos. So we're trying to think of some ways to make sure that we're participating. That's the end of this video. Stay tuned for more. Keep up to date with stuff. Email me if you got questions, because I'm sure there are going to be a lot of questions come up, especially that first week we're back in action, but in a very different way.